Okay, today I'm going to be talking about avian disease ecology and some anthropogenic implications. So I'm going to be giving a brief introduction, going over some pathogens in commercial species, some pathogens in wild species, and talking about some urban effects on those pathogens. So birds are super widespread, appearing on every continent on the planet except for Antarctica. And due to this ubiquitousness across ecosystems, they are sort of keystone species across the globe. They're important for agriculture, tourism, they're food sources for numerous different communities worldwide, and they're important for even more than that. So, because of this vital importance, it is so important to understand the pathogens that can affect them and how those pathogens can spread throughout these different populations. So, looking at some pathogens that affect commercial species of birds like chickens or ducks or turkeys, we start off with Newcastle disease. Newcastle disease is a single-stranded RNA virus that comes in either a lysogenic, mesogenic, or villogenic form, meaning there are three different forms of this virus of three different sort of virulences and pathogenicities, with the villogenic virus being sort of the worst to come down with. Newcastle disease spreads through direct contact, meaning healthy birds have to come into direct contact with either an infected individual or their infected droppings or nasal oral secretions from an infected individual. Because of this, Newcastle disease is density dependent, meaning the more birds you have together in an area, the faster this is going to transmit, which is not great for these giant industrial farms where thousands of chickens are very, very closely packed in together. Newcastle virus can be ID'd by PCR or ELISA testing, and some symptoms of Newcastle disease include depression, diarrhea, paralysis, and even death. Moving over to Merrick's disease, I just really like this picture, and I'm really happy I get to talk about it. Merrick's disease is caused by an alpha herpes virus, and there are species-specific strands of the virus that have different pathogenicity. For example, the chicken-specific strand of the virus is more harmful than the turkey-specific strand of the virus. Because it is a herpes virus, it is a lifetime infection, meaning just like chickenpox or shingles, once a bird gets it, it's stuck with it for quite a while. Merrick's disease is transmitted through contact with feather dander. So these little dust mites that are on the feathers of these birds is where this virus likes to hang out. It is highly contagious because this virus is able to survive on that feather dander outside of the host for up to a month, meaning if it's in the environment, it's going to stay in the environment for quite a bit. It also, like Newcastle, can be ID'd through PCR or ELISA testing. And some symptoms of Merrick's disease include inflammation and tumors in the nerves, spinal cord, and brain, which are these white lumps we see in this image to the right. These are all the tumors caused by Merrick's disease, which cause paralysis, and it can also lead to impairments in vision and movement in the affected birds. Good news is chicks post-hatching can be vaccinated for Merrick's disease. They can be vaccinated against these herpes virus, meaning there is some positive steps forward for the control of this virus. Now, moving over to a few pathogens that are very common in wild species of birds, we start first with avian malaria. Malaria is a protozoan parasite of either the Plasmodium or the Hemoproteus genera. They attach to red blood cells, just like we see in this image to the right. All of these ovular shapes with dark purple nuclei are avian red blood cells, and these light purple structures attached to them are the malaria parasites. And malaria is spread, as you might know, by invertebrate vectors, mosquitoes for the Plasmodium genera, or Hemoproteus, or biting midges, which spread the Hemoproteus malaria. There are theoretically, it's been proposed, as many species of parasites as there are species of hosts, which goes hand in hand with the diversity of avian species I talked about earlier. Just like that, there are malaria cases in birds found on every continent across the world, except, of course, for Antarctica. And avian malaria can be identified through blood smear analysis, like looking at images of blood cells like this and identifying the malaria parasites within those blood cells. Symptoms of avian malaria can include anemia, lethargy, loss of appetite, and weight loss. And finally, coming to one that's sort of a big ticket right now in avian disease ecology, we have avian influenza, which is a RNA influenza virus, and it comes in two categories: highly pathogenic avian influenza or low pathogenic avian influenza. It is transmitted through direct or indirect contact, either direct contact between infected and healthy individuals, or more indirect method by contact with infected materials or infected bodily fluids. Symptoms of AI can include decreased egg production, misshapen eggs, diarrhea, lethargy, sneezing, and death. There is potential for spillover, as some studies have shown avian influenza being able to infect mammalian hosts like foxes and seals, which sort of makes me anxious at the possibility of spillover into humans. And to wrap things up, I have a couple of papers here, a few studies looking at how a couple of these pathogens, how they fare in urban versus rural environments. This first one, entitled Effects of Land Use Change on the Diversity and Epizootiological Parameters of Avian Hemosporidia in a Widespread Neotropical Bird. So here we're looking at malaria parasites in the common bush tanager, this beautiful bird on our right here. This team took blood from these birds across an urban-rural gradient, and they then took that in the lab to test for genetic diversity of these malaria parasites, and they found that the diversity of these parasites decreased in more urban environments. So there were less species of parasites in the more urban environments. This could be proposed, it is proposed that this could be due to a lower diversity of hosts and vectors, or because of environmental toxins in the urban environment sort of limiting the spread of the parasites. The other paper I have is entitled Avian Influenza A, Virus in Wild Birds in Highly Urbanized Areas. Here we're looking at wild waterfowl, like ducks, geese, and gulls across a similar urban-rural gradient. This team took blood samples as well as cloacal and oral swabs from these birds and looked at PCR testing for influenza viral particles in these samples, and they found that there were fewer positive cases in heavily urbanized areas. And it is proposed that this could be due in part to larger aggregations of birds in rural areas versus in urban areas. So that's what I've got for you today. If you're interested in anything I just talked about, please dig into any of these literature and papers I've posted here. So thank you.